Welcome to the Bioinformatics CRO podcast. I'm Grant Belgard, and joining me today is Yuri Deigin. Yuri, can you introduce yourself, please?
Sure, yeah. Thank you for having me on the podcast. I'm Yuri. I'm a drug developer. I'm leading a longevity company called YouthBio. We are developing in vivo partial reprogramming gene therapies to ultimately create rejuvenation therapies, but in the interim, create therapies against existing age-related diseases like Alzheimer's, for example. Yeah, happy to chat about any topic today.
Yeah, I'd like to learn more about YouthBio and how related do you see the underlying causes of various age-associated diseases?
All right.
How much convergence do you think there is?
Yeah, right into it. So yeah, I guess YouthBio is really the kind of culmination of, most importantly, my understanding of how aging works, and that is epigenetically driven, and that, yeah, obviously all age-related diseases in my mind are caused by the underlying process of aging. And of course, different diseases could be different manifestations of the same process, or different results of this process of gradual epigenetic slowing down of the systems, repair systems, and all other systems that help maintain homeostasis. And so reprogramming is one way to actually intervene on the level of epigenetics, because, well, reprogramming just epigenetically brings the gene expression pattern all the way into the embryonic-like gene expression pattern, and that results in all of the changes associated with, cell morphology and function, ultimately, as it's, of course, driven by gene expression, but it all starts in changing the gene expression pattern. And what was noticed that during the reprogramming process, the cell is also rejuvenated both physiologically and on the level of its transcriptome, basically on the level of gene expression. In the initial stages of reprogramming, it seems that the pattern of the gene expression of an old cell gets shifted closer to a pattern of gene expression of a younger cell, of the same cell type. And so this is really the kind of the foundation of partial reprogramming, where we try to use this process of rejuvenation during reprogramming and stop it at that precise moment where the cell still remains of, the same cell type that you start with, but it gets rejuvenated to some degree. And we don't want, of course, to proceed further. We don't want the reprogramming process to proceed further, because that could make the cell lose its function and lead to all sorts of problems. So that's the partial aspect of partial reprogramming. And basically the underlying hypothesis that aging is epigenetically driven really synchronizes well with the approach of reprogramming, which, as I mentioned, works on the level of epigenetics. And that's why I think it's a very powerful paradigm if we can figure out how to do this safely in all sorts of different cell types, because I think it actually is necessary. Like, different cell types necessitate different approaches to using reprogramming in those cell types. Then I think we can have very powerful instruments to rejuvenate those cell types. And so eventually have a systemic therapy where you can target multiple cell types, multiple organs, and eventually obtain this, holy grail of systemic rejuvenation, and make aging either slow down or even reverse aging to some degree in healthy people. That's the ultimate goal of longevity research, to make healthy people healthier for longer, make aging slow down in healthy people, and radically extend lifespan and health span using such approaches.
How do you think about aging fundamentally, and what is the relationship between that and the epigenomic changes that are associated with aging?
Yeah, it's a very interesting question, because, just recently we have had a discussion on what is aging for a while. I think it's been spearheaded by Vadim Gladyshev. And just recently we had another installment, his talk on the topic. So it's actually interesting that there isn't a consensus, or at least a complete consensus in the field on what aging is exactly, because different people call different things aging. And obviously, there's some common things that we observe that realize is what aging is associated with. We all know aging when we see it, right? We know an old person from a young person, and like even non-experts in gerontology, lay people, obviously are very good at telling apart an older person from a younger person. So obviously we have that really actually deep down in genome recognizing aging and recognizing old individuals from young individuals, even across species, right? So this is obviously something very fundamental to life, to, well, at least, I guess, mammals and higher animals, recognizing old from young. But on the level of gerontology and biology, I think, to me, aging was just the process that causes mortality risk to increase with time. So basically, if your mortality risk does not increase, to me, that means the organism is not aging. So if the mortality risk stays constant or actually decreases with age, then that's an example of either a non-aging organism or a non-aging stage in the life history of an organism. For example, I think between ages like 5 and 8 or 5 and 9 in humans, the mortality risk actually decreases and reaches like a minimum between ages like 8 and 10. And then after puberty starts, then that mortality risk starts to increase. And it keeps increasing exponentially until we die, with potentially a plateau between like age 20 to 30, 35. And some gerontologists actually argue whether there is a plateau or there is a slowdown or there isn't, or just an aberration or statistical aberration. But basically, this is just, I think, one key observation or definition even of aging. Basically, aging is the process that increases mortality risk. And in humans, of course, it doubles mortality risk every 8 years. So it's an exponential increase. And in other animals, it's not necessarily so. In some animals, actually, mortality risk decreases with age. There's examples of turtles, for example, who seem to we observe decrease in mortality risk and actually increase in their fertility in some species of turtles. But of course, there are other ways to define aging as, for example, accumulation of various damage, intracellular, extracellular, or other manifestations, other hallmarks that people associate with aging. And they call, that is the aging process rather than, the downstream effect of increased mortality. So you can, I think, as any academic field, you can have a lot of debate about the precise details. Ultimately, circling back to what we were starting to discuss in the beginning of this question, we obviously know aging when we see it. And we would know rejuvenation when we see it. If you're able to take, a 40-year-old person and rejuvenate him or her to look and act and physiologically be at the level of a 20-year-old, or same, pick any two numbers, obviously everybody would agree that that is true rejuvenation. And you can do this, of course, in other animals as well. Mice, for example, too. Specialists in mice would obviously see rejuvenation if you take a 2-year-old mouse and rejuvenate to a little 1-year-old mouse. There won't be any question that this is actual rejuvenation. So from that standpoint, you can have, and obviously there's other biomarkers of aging. I think right now there's physiological biomarkers of various functions of the organism, like lung function, kidney function, heart function. There's also epigenetic biomarkers are built on the known data of health status, mortality, and all sorts of different ways that you can approximate the aging process and evaluate whether you're able to observe a slowdown in the aging process or actually reversal of the aging process if you see, for example, biological age being reduced after some interventions. So, sorry, I think I'm starting to maybe go a little bit off.
No, this is good. I mean, it kind of raises, I think, a natural question. How do you think about aging and biomarkers of aging in the context of drug development, right? Because certainly different stages, you would need to consider different things. And how does all that tie into the question of what is aging? Is that question essentially more of an academic debate, or would it really impact at a fundamental level what your primary endpoints would be in a clinical trial?
I think we have enough at this point, enough biomarkers to assess to a pretty good degree any interventions that claim to rejuvenate or slow down aging, precisely because we have different biomarkers and we can test function and we can test epigenetic age. So from that standpoint, I don't think there's anything limiting drug development or therapy development for rejuvenating therapies, especially because at this point and in this regulatory framework or regulatory landscape, you really need to go after interventions, after diseases. Interventions have to go after diseases. So it's not like you can have clinical trial of a healthy person taking some sort of rejuvenation therapy and then measure endpoints. You really need to have some patients that you administer the therapy to, and then you evaluate clinical endpoints and biomarkers that are improved by that therapy. So from that standpoint,
what indications do you think would be especially promising for that as a gateway to ultimately develop drugs that more generally target aging? How would you think about starting?
Well, I mean, we have example of senolytics and osteoarthritis of the knee, right? People thought that would be a good indication to try. And I think there's different ones that you can go after. And well, with the TAME trial is another methodology where you have a whole basket of age-related conditions and you have people kind of susceptible, older people to those conditions. And you then evaluate whether your therapy, your drug, metformin, I guess in that case, is able to reduce the incidence of a number of age-related conditions. That's one way to actually measure the effectiveness of a potential therapy. And other therapies, I think, depending on the actual intervention, they could have different optimal targets, optimal diseases, which could be the first ones to go after in a clinical setting. I mean, reprogramming is, like if we're talking in the context of partial reprogramming, I think it has a whole host of different areas in which you could demonstrate benefits in that particular disease model or actual disease or predisposition to disease, where you can confidently say that not only is this helping this particular disease, it could also have a systemic effect if you're able to target multiple tissues. In the context of reprogramming, I think skin people think is a low-hanging fruit because you can have very quick results, visually observable improvements. And that's why I think a couple of companies are going after skin, are going after dermatology, and also other areas in terms of hematology and immunology, people are also considering. But also, I think in the context of the central nervous system, brain diseases, if you're able to demonstrate using approaches of partial reprogramming, even a slowdown in the onset of various neurodegenerative pathologies like Alzheimer's, like Parkinson's, etc., then it's a very compelling case to be made that this is truly a therapy that's at least slowing down the onset of aging in this particular tissue, the onset of age-related symptoms of this particular disease. And also, if you're able to reverse the symptoms, that's a much more stronger a case that you're actually rejuvenating the underlying organ. So I think, while they're very neurodegenerative diseases, they're obviously hard nuts to crack. I think the upside in their cases is really high. So there's huge unmet clinical need. In Alzheimer's, for example, you're able to demonstrate clinical benefit there. I think it would be a very compelling case for people to take notice that this approach is a very strong rejuvenating approach.
So I think neurodegeneration is a really good use case because, we do know there is some degree of genetic overlap, several neurodegenerative diseases, and also, gene expression signatures associated with age and then QTLs for those and so on have been identified in the context of brain age, right? What concerns me a bit, though, about the paradigm of slowing aging overall and having a substantial impact on the rate of age-associated disease is if there were a single underlying factor of aging that kind of affected, overall physiology in a similar way, something analogous to G and intelligence or whatever. You might expect to see that in genome-wide association studies of different age-related diseases in different organ systems, and to have, some of the usual suspects coming up time and time again. And within certain classes of diseases, of course, you see some of that. So neurodegeneration, tau, for example, comes up quite often. But you see very, very frequently, which you would expect if you have age-matched controls versus, people with this disease or that disease. On the other hand, of course, there are many facets of age associated biomarkers that are strongly correlated with one another, right? So these frailty indices and so on. And of course, we do have genetic associations with measures like that. So do you think about it as, okay, we will identify treatments that are effective for maybe somewhat broader classes of diseases in kind of, reducing their rate of onset? Or do you think there is a good possibility of slowing everything down together?
I think theoretically there is a good possibility of slowing things down together. I think aging is centrally regulated, to be honest. I mean, I think actually it's a program, and I know I'm a minority opinion on this because, I mean, there's so many life stages that are definitely centrally regulated, and we have to have everything in a very coordinated concert to be developing just, at the right time in the right proportions to have an embryo develop properly, and also to have a puberty onset at the right time and all organs start growing at the right time. So there is obviously a central synchronization mechanism. Now, we still haven't figured it out how exactly it occurs. And well, there's some hypotheses that, it could be the hypothalamus could be one such controlling master regulator of aging, because it is actually controlling a lot of daily, monthly processes, or maybe even on a longer time scale. Obviously, during embryogenesis, it plays a very important role. I mean, that said, I think this is a parallel search for this kind of master synchronization system. And also, of course, I think if we're able to slow down development, that we've seen that obviously slows down aging, or actually puts aging on pause if we're able to delay the onset of sexual maturity. And actually, some people are pursuing that as a rejuvenation strategy. There is a gerontologist in Ukraine, Valery Golub, who's studying this in rats and reptilia, trying to see, because obviously we've seen in many organisms that neoteny greatly extends lifespan, and also experimentally, if you're able to pause development or slow down development, that also extends lifespan and seems to, put aging on pause. But coming back to the partial reprogramming approach, it's a bit of a different way of going about it. It's just targeting key organs that are playing, the biggest role in our age-related pathology. Of course, heart disease being one of them, and other organ systems, and hoping that we can target enough key organs and key systems that result in a systemic effect where we can slow down aging of the entire organism enough to, produce sizable gains in health span and lifespan. But also there is a hope and a hypothesis that we're also exploring, that maybe by modulating in the certain areas of the brain, rejuvenating them, maybe we can then achieve systemic effect. But obviously that's just a hypothesis that we need to explore. But, to answer your question, I do think that it might be possible to have an intervention going at the entire aging process, or at least the actively driven aspect of the aging process. Because obviously, after some time, there are also stochastic things happening that are not part of the epigenetic landscape being driven, but they're kind of side effects of genes allowing stochastic changes to produce enough damage that it actually starts to accumulate. But that's a bit of another tangent that I'm happy to explore.
I think there are maybe a couple tangents we'll go off on in a bit, but I think it'd be really interesting to hear kind of about your journey. How did you end up here? What drove you in the direction of science, etc.?
Oh, yeah. My journey to longevity started with just regular drug development and meeting people who were in the same drug development ecosystem, but there were also longevity-minded people who just shared with me the idea that aging is the causal factor in age-related diseases that I was working on, they were working on, Alzheimer's, for example. And basically saying that we can and should try to intervene in aging and slow aging down and ultimately, hopefully, reverse it. And that was a revelation at the time to me because, as most people, unfortunately, on the planet, I never thought that aging is something that needs to be intervened in or should be, or is it possible to be intervened in. I thought, aging is inevitable, just kind of everything ages, and didn't even, think that there is something that could be done about it until my eyes were opened by my friends at the time. And obviously after, they made me aware of this whole industry and research community looking into aging, fundamental mechanisms of aging, and trying to intervene in aging, that very quickly I realized that it makes all the sense in the world. That yes, obviously aging is the causal factor in all those diseases, and there's no good reason why we should settle for the lifespan that we inherited biologically from our ancestors. And we have the intellectual capacity to analyze biological process and finally have the tools to intervene in biological processes, that it only makes sense for us then to turn our intellect and our abilities to intervene in biology to the biggest limiting factor in our lives, which is aging. And very quickly, I became a very passionate activist at first about aging research, about the necessity to research into aging, and just trying to open other people's eyes, just like my eyes were opened by very, very simple and logical information, and just making people aware that, there is a thing like aging research out there, that it's possible to intervene in aging. It's possible to extend lifespan in model organisms. It has been done many times. And it's only a matter of time until we find effective interventions that can be applied to humans and produce sizable gains in both health and lifespan. And eventually I was able to then not only do the activist part, but do the drug development related, or start doing drug development related to longevity. And that came in the context of partial reprogramming. Because at the time when I learned about partial reprogramming, I was already of the mind that aging is driven by epigenetics. To effectively intervene in aging, we need to intervene at the level of epigenetics. If we're talking about already formed organisms, of course. If we're talking about new organisms, probably we can intervene at the level of the genome, but unfortunately we're already cooked organisms using the cooking instructions stored in our DNA. So if we want to change something in our biology, we have to go after gene expression. And as I mentioned in the beginning, partial reprogramming is one way to intervene at the level of epigenetics, at the level of gene expression. And it's a very effective mechanism because it uses an underlying process that's already fortunately encoded in our genome to do this, to restore gene expression of the primordial embryonic stem cells. And it's a very powerful tool that we can use to essentially hack for our purposes to rejuvenate adult organisms, adult cells. And I started on the path to try to translate this paradigm into therapies very early on, back in 2017. The paper came out in December 2016, and then I founded the first company, Ethereum, dedicated to translating this paradigm in the summer of 2017. So I got really excited about, because it was fell in fertile soil in my mind, the partial reprogramming paradigm, because I already thought that epigenetics is the driver of aging. So again, I took, a little longer maybe than I should have to answer your question. But happy to dive into any details of the journey or any particular aspects of it that could be useful to other people.
Yeah. I think it'll be interesting to hear about, the lessons you learned through that. But before we get into that, to go off on a bit of a tangent, you were obviously quite early to this, and of course now partial reprogramming and aging research is much more mainstream than even 5-6 years ago. But you were also very early on the important aspect of coronavirus, right? Can you tell us about that, and kind of how did you get there? I recall reading your very, very long blog post years ago, and I was really impressed. what was kind of the impetus behind that, right? I mean, because at the time that was very, again, not mainstream, but it's one of those things that I think has become much more mainstream in the in the years since, in large part because of kind of what you kicked off. So
Yeah. It was just another tangent, essentially, that I got curious about the origin of the coronavirus, COVID, I guess, not just any coronavirus, but the coronavirus that caused the global pandemic. Yeah, I basically, like everybody else, was curious in early 2020 where the virus came from. And initially, a lot of people made the obvious connection that, Wuhan has the premier lab in the Wuhan Institute of Virology that has been studying coronaviruses, but in the very early stages of, the pandemic, this making the hypothesis that the lab had something to do with the outbreak was considered a crazy conspiracy theory. And powerful and very respected scientists were putting their names and their reputation behind this to dispel this conspiracy theory. And initially, I just trusted them. I thought, hey, if Nature Medicine says that the virus must be natural, and there's like 5 or 6 virologists, and then Anthony Fauci saying, that they are to be listened to, then they know what they're saying. And but I at some point just decided to dive a little deeper, and instead of just outsourcing my thinking to them, decided to try to see for myself from first principles. Does it make sense? Does conspiracy theory make sense or not? And so I read the Nature Medicine paper, and I was a little disappointed by the logic, because once you kind of strip out all the virology lingo, you're left with very unconvincing logic that basically it's the absence of evidence that the authors are trying to sell as evidence of absence, evidence of no possibility that this could have come from a lab. And then I started digging deeper, and I saw that the kind of research that they were doing in Wuhan was exactly potentially similar to what could have produced this coronavirus. And
of course, this was long before the diffuse proposal had come out and so on.
Oh, yeah. Diffuse was a year afterwards. And basically it was just circumstantial evidence of, look, I mean, this is clearly a huge red flag that the lab that's been collecting coronaviruses from all over the world and manipulating them in all sorts of ways had the outbreak of this coronavirus with a very unusual feature, a couple of unusual features, one of them being this furin cleavage site. And it was very suspicious to a lot of people. And basically, I wrote this up in a blog in like early 2020, published it, and then it kind of took on a life of its own and got me kind of deeper into the rabbit hole and into the community of people, like-minded people who were also very suspicious that this could have come from a lab, and we're continuing to investigate all aspects of everything surrounding basically the outbreak, the work in the Wuhan Institute of Virology and other EcoHealth Alliance as well, of course, what their involvement was and how they were using grant funding and their close cooperation with the Wuhan Institute of Virology and other labs to essentially collect and manipulate viruses in the lab to make them more infectious for the purposes of assessing how dangerous this would be if it happened in nature. Basically try to second guess or try to guess what nature could do to make viruses more dangerous for humans. And they did this with SARS-like viruses. They did this with MERS-like viruses, MERS being the Middle Eastern respiratory virus that is very deadly, is like a 37% fatality rate. And they did a lot of research on that in the Wuhan Institute of Virology together with EcoHealth Alliance as well. We learned that, a year or two, well, not two, a year and a half after, of course, the initial outbreak, because of all the Freedom of Information lawsuits that were filed to NIH and other agencies to actually see what has been going on in this kind of research between EcoHealth, NIH, who funded a lot of these studies, and the Wuhan Institute of Virology, who were doing a lot of these studies. But I, again, diverged. But basically, once I gotten interested in this origin of coronavirus, the community of other people were also, that I got together involved with, we started calling ourselves Drastic. It was a Twitter-based group of activists trying to investigate the origins of the coronavirus, and we investigated some additional aspects, published a bunch of papers on very suspicious discoveries. And basically, at some point, yes, the diffuse proposal was leaked by Drastic. The diffuse proposal being, of course, the joint grant proposal by EcoHealth and the Wuhan Center of Virology, in which they pitched to DARPA this idea of collecting novel SARS-like viruses and also engineering novel furin cleavage sites in SARS-like viruses. Because up until now, and actually never since, SARS-like viruses never had a furin cleavage site at that spot, and it seems actually to be evolutionarily discouraged in bats, because in bats, these viruses are gastrointestinal viruses, and furin cleavage sites actually seem to be detrimental to the viruses being tropic to the GI tract or something. Basically, furin cleavage sites, for some reason, make viruses more preferential to the respiratory system. But of course, for humans, that's the biggest way how viruses get transmitted through once they're airborne, through the respiratory system. So for a virus to jump from bats to humans, the kind of key skill for it to pick up is to actually become tropic to our respiratory tissues and cell types. And inventing or getting a furin cleavage site actually is a catalyst for a virus to do that. It basically turns the virus from a gastrointestinal virus into a respiratory virus, and that makes it very high threat for transmission. It makes it so much more transmissible for humans especially, right? And in the context of research on coronaviruses, it's been very well known that furin cleavage sites expand tropism of such viruses and make them actually respiratory viruses. And it's been experimented with other virus types, coronavirus types, to engineer novel furin cleavage sites and observe how that changes the tropism or the preferences of the virus to different cell types and different systems, including the respiratory system. And so in that diffuse proposal, one of the things they described to put in their experimental roadmap was to try introducing novel furin cleavage sites, basically to assess the risk of how likely it is in nature for a furin cleavage site to arise, and to also model what would happen if a particular virus got a furin cleavage site, how likely would it then be to jump to humans. And of course, the SARS-CoV-2 virus has this receptor binding domain that's very highly preferential to human ACE2 receptor, which is very, very interesting characteristic. It scores like number one on the list of all different animal ACE2 receptors to be preferential to the human ACE2 receptor, which is, very odd for a bat virus. But maybe there's a species of bats who has a very similar, or an ACE2 receptor that's also very high binding affinity to that particular conformation of the spike protein on the SARS-2 virus. And of course, not only, does the spike protein binds well to our ACE2 receptor, the furin cleavage site makes it so much more transmissible and so much more preferential to our respiratory system. And so once the diffuse proposal was publicized in the summer of 2021, to a lot of people, this was a very convincing additional nugget of evidence
that was what pushed my posterior probability on this way over 50%. I recall reading through it and saying some words that would not be appropriate for a family podcast. It was just shocking. And I was certain at that point that, these surely global regulations on gain of function research would be coming down swiftly. And the amazing thing is, even though later that summer, we got to the point where most Americans believe, WIV was the origin of the virus. And, of course, former CDC director during the outbreak has, said something along those lines. Obviously, a lot of people within the U.S. intelligence community were concerned that was the case, and several people in Congress, right? So one would think something would have been done about it by now. What's shocking is, there's probably more manipulation of coronaviruses happening now than before, right? it's extraordinarily dangerous. I mean, even if you don't buy that it, came out of WIV, like surely even without that, just knowing how common it is for viruses to escape from laboratories and so on, the cost benefit of this kind of work is so overwhelmingly on the cost side and not the benefit side, right?
Yeah, for sure. The SARS-CoV-2 escaped definitely once from the Taiwan BSL-3 laboratory, and there were actually acknowledgments, like in internal emails, that before that it escaped within a Chinese laboratory. And the first SARS virus that was much less transmissible escaped like 4 or 6 times from different labs. So obviously, coronaviruses obviously can escape from even high level of security biosafety labs. So, and yes, and obviously, I'm sure there's countless times where people were infected handling coronavirus samples these days, because so many labs now work with the coronavirus, and they just obviously it's so hard to track now whether the infection you got as a researcher is from a lab or just somewhere outside. I mean, in Taiwan, they could track it down because essentially it was very low circulating SARS-CoV-2, if not zero at that point. So, and they were able, I think, to do genomic tracing to confirm that this particular sample is the one that infected the researcher. But yeah, also like all sorts of potential gain of function research that people have been doing or are doing now with the COVID virus, SARS-CoV-2. One of the earliest ones was these Italian researchers in Siena, I think, in the beginning of 2021, they passaged SARS-CoV-2 in the presence of neutralizing antibodies from like people who had COVID or were immunized, to actually see how it could evolve to escape, those immunities, escape that antibody immunity. And they succeeded. They created a whole new strain that was very good at infecting people who already had immunity to the original SARS-CoV-2 virus. So, if that escaped, we could have had a whole new episode of the pandemic with a whole new strain. And what happened with Omicron, to me, is highly reminiscent or highly similar to what, could have happened if a gain of function research could have produced a strain, then just got out and infected people and started circulating. Because Omicron is just so different from its last known ancestor, it has like 30 spike mutations, basically, like that it developed seemingly without any intermediates. And like to this day, it's still a mystery. How did Omicron arise, and where was it circulating between like June or November of 2020, when its last ancestor was seen, in November or December 2021, when Omicron emerged? Where was it all this time? And the idea that it wasn't just some one immunocompromised patient in which it could have developed those 50 mutations, including 30 spike mutations, to me, it's a very implausible hypothesis, because we have been observing quite a number of immunocompromised patients, and in none of them did we observe such a huge number of mutations arise. There's usually a couple of dominant ones arising and then maybe a few others, but I think the most we've seen is like 10 mutations in the spike of a person who had like 18 months infection present for a duration of 18 months that they couldn't clear. And so, but now in Omicron, seemingly between a year since seeing its last ancestor, developing 50 mutations with 30 spike mutations, it's really, really crazy. So yeah, it could have been a lab leak as well, and who knows what other gain of function research is going on, because obviously it's not a regulated space, and even if you regulate it in one country, who knows what happens in countries that are not signed on to be parts of some regulations. So it's a huge, huge existential level problem for us, for humanity, that unregulated gain of function research of pathogens going on in all sorts of labs all over the world could lead to pandemics, maybe even worse than this one. I hope not.
It's just wild to me, right? Like when I was reading the diffuse proposal, one of my immediate thoughts was, this is kind of the Manhattan Project movement for the life sciences, except far more people have been killed by this than have been killed by nuclear weapons, right? So I was expecting surely there would be
And that diffuse proposal was actually much broader than that. They proposed actually creating vaccines for bats with which they would go out into the field and immunize bats preemptively. And who knows what wrong could have gotten from that approach, and what maybe new combinations of viruses bats could have developed due to that. Thankfully, diffuse proposal was not funded, and that particular aspect of it, of trying to actually go out and change ecosystems with preemptive vaccines in wild animal populations, this was criticized by the reviewers. But of course, the molecular biology experiments, even if they weren't funded by DARPA, they could have been funded by many other agencies, including Chinese funding agencies, or even you don't need a lot of funding to do genetic manipulation of viruses. If you already have the viruses and you've collected some, and to insert 12 nucleotides into a coronavirus genome doesn't really take a lot of money or a lot of time for your postdocs or PhD students.
So I mean, why do you think there hasn't been a reckoning on this, right? Most people believe this is what happened. Plenty of people in power have said this as well. And yet there hasn't been an aggressive effort to shut this down.
To shut down gain of function? I mean, yeah, it's very hard, I think, to come up with novel global regulation of an entire field of virology. If we're talking about pathogen research and classifying what exactly is gain of function for a pathogen and what isn't, and in what context it should be permitted or should it be completely banned, and what about places that don't sign on to this convention. Virology is just going to migrate, or virology labs will just migrate to those geographies that don't have that regulation. Of course, if you ban it in the United States and Europe, that's the major sources of funding. So I think that could lower risks of various, pathogen gain of function research ongoing, and then greatly decrease it in number. But
I guess the challenge is, it's much more inexpensive and widely accessible to do this kind of work than it is to enrich uranium and build nuclear weapons. Right?
Oh yeah, these days, absolutely. Biology, I think from an existential risk perspective, is much higher on the list than nuclear war or nuclear terrorism, because bioterrorism by comparison is much cheaper, unfortunately. Hopefully, we won't have bioterrorists listening in and oh, okay, I was enriching uranium in my backyard. I think I'll go build a virus lab now. But I think the process is actually ongoing. It's slow, and we see some of it starting to happen with now the Republicans having these hearings, and maybe they will result. There's two questions. One is about the origins, which is of course important, but it's not even if we don't find out the exact origin.
Right. Even if we're not certain, I mean, there's certainly, I think it's undeniable that it's a very plausible source. And even in light of it being a plausible source, it very clearly is something we need to button up and make sure that kind of thing does not happen. Or even if you say it didn't happen the first time, that it doesn't happen, right, in the future.
Right. I mean, not just the SARS leaks I mentioned happening in the past couple of years. And just before the Wuhan outbreak, there was a brucellosis outbreak in China, in Kunming, I think, in Yunnan, in the lab, and 100 of people got infected by. It's much less transmissible, thankfully, bacterial infection. But still, obviously, it's a lab-borne infection. So these things happen, and these things are dangerous. It's just a matter of the right pathogen escaping that could be so much transmissible that it could cause a pandemic versus something that isn't. But absolutely should be taken seriously. And I think also the problem is that politicians are blissfully ignorant on where biology stands right now. And when they find out, their hairs stand up. How easy it is to manipulate biology and how cheap it is these days, and basically what Wild West, to quote Jeremy Farrar, exists in all over the world in labs. Basically, nobody is regulating what you can do in the lab. If you want to supercharge a virus, nobody's going to, the virus police won't come and say, "Why did you do this? Didn't you realize that this is dangerous?" So hopefully soon enough, people in power will come to understanding of what's going on and what's possible and what's dangerous.
If someone is just, hell bent on doing it, it would be difficult to stop them. But certainly the vast majority of the work ongoing where something like this could accidentally get out and cause lots of problems. It's funded by funding agencies. People are doing it to publish work that's peer reviewed. If there were a common consensus, if scientists just got their act together and did this even without any kind of government intervention, that would dramatically reduce the likelihood. But that doesn't seem to be happening.
Yeah, exactly. And I think the incentives are misaligned here because in order to publish something, in virology, you have to create, interesting research. And right now, it's very hard to do interesting stuff without genetically manipulating viruses or, modeling some different changes that could make viruses more pathogenic or more transmissible, I think. So, but at the same time, from the benefit of society or humanity as a whole, this type of research, supercharging viruses or creating more transmissible viruses or more dangerous viruses, I think has a very, very little benefit, if any. But at the same time, the risks are huge, and the risks are obviously shared by the entire world. And so, if we're worried about just one bioterrorist, it's one thing. But if we're worried about, thousands of virologists doing this thing and making some mistake, or not even a mistake, or just a supercharged virus could escape, even if they follow procedures, maybe some equipment malfunction or something happens of that, people don't anticipate that could lead to pathogen to escape, then, the probability of this, if 1,000 people are doing it versus just one crazy terrorist is doing it, I think are much higher. But it's a different conversation.
So I guess coming back now after, I think it was a pretty interesting digression in your own career, kind of a side project. Yeah. So if you could go back several years before you started the founder journey, what advice would you have for yourself?
Oh, that's a tough question. Invest in Bitcoin, maybe. That'd be one advice. If we're talking something useful for people these days, right, who are entering longevity, like in terms of career advice, I think now is a much better time because there's all sorts of fellowships and programs like, LBF, Longevity Biotech Fellowship, available for people who are just getting interested in longevity and want to quickly orient themselves. I think the number of resources available is really large compared to like a decade ago or more when I was entering the field. So just the advice would be to study existing, kind of description of the landscape of the field and various areas of research, and just, I think, not be afraid to reach out to people and ask for advice. If you're interested in career switching, there's all sorts of companies open to people coming from different fields. And obviously, if you want to do this, if you're interested in longevity, one advice I would give is not to defer it, not to procrastinate, but to get into it as quickly as possible. Because I think the more you get drawn into a career, the harder it is to then to switch over. And longevity, I think right now is at a good point where you can enter into a lot of new companies or even labs on the academic side. And the tidal wave that's coming, it's going to lift, I think, your career and your progress in this field as well. So maybe, just don't delay.
Yeah, it's interesting because, you had a somewhat circuitous path, right? Where you were in tech for several years before getting into the pharma.
Oh, yeah. Before drug development, I was in tech. My first career was in tech, and I did a computer science degree in mathematics, and I worked for IBM. I did a startup actually in mobile applications back in like 2002 before it was cool. But I pivoted then into drug development. I wanted to do drug development for a while, and actually, the time has come back like in 2008 for me to do this. So I decided to do an MBA to help me get into the pharmaceutical business development. And that was the key pivot. But then to get into longevity drug development, that took me also some time before an opportunity came, which, yeah, I think was a perfect opportunity with partial reprogramming because it was just the right approach for my current and then understanding of aging as an epigenetic process.
Well, I guess in terms of getting in early, you're 2 for 3, right? So longevity field, the COVID origins, but not Bitcoin.
Oh yeah. There's a lot of other investments that, if you had hindsight, would be nice to know about back in a decade ago.
Cool. Is there any advice you would give to people who are launching their first company?
Network. I think, yeah, networking is really, I mean, but obviously it's pretty obvious advice. And maybe, yeah, it's don't get discouraged because it's very easy as an entrepreneur. There's always a roller coaster. There's always going to be, down times where you think, everything's going badly, but you just have to kind of keep going and keep the faith that eventually there's going to be another positive development that will, and just, yeah, like longevity in terms of being entrepreneur is also very important. You have to keep doing what you're doing. And if you believe in it, of course, of course, if something changes and you realize that the business model you had was wrong, or the therapeutic approach you were exploring doesn't really work out, then of course you have to change. But if all the fundamentals remain the same and it's just kind of the environment is not really good, like right now, economy is not great. So a lot of pundits are saying there's going to be like an extinction of startups coming, but you have to kind of close yourself to the noise and just keep doing your job and just, yeah, try to achieve your milestones that you put forth for yourself and for your company. I don't know. That's probably the same advice everybody gives, but
No. I mean, it's good to know what people emphasize because there aren't enough hours in a day to do everything one is advised, right? And so even hearing the same thing time and time again, you get extra votes for the importance of that. Yeah.
yeah. I think everybody says resilience is a key attribute for entrepreneurs, and that's true. And
get punched in the face and
yeah, exactly. But it's true. You have to keep going in the face of adversity, and of course, also the more you do it, I think the better the thicker your skin becomes, and the better you become at managing it. So also, yeah, I think for younger entrepreneurs, it could be a little tougher, but it's just something that I think, like with anything else, but the time and practice, you get better at it. So
develop thick skin.
yeah, stick to it. Don't get discouraged. And also, yeah, I think it's also important to have people who can help you both emotionally and like with advice, and like support groups are important. And that's why I think also within the fellowships, like the ODLB fellowship or the longevity biotech fellowship, these mastermind groups, as you know, that could be very helpful because there you can connect with people going through similar things, similar entrepreneurs or scientists, researchers, or postdocs going through similar adversity, and connecting with them and getting support, both emotional support or just some advice or some other help. I think it's really important to have that kind of support structure, support network for everyone, entrepreneurs and academics alike. So yeah, get that support structure in place, network and try to form this group of people who can support you through the tougher roller coaster rides.
Solid advice. Thank you so much for joining us, Yuri. It's been a pleasure.
Thank you. Yeah, well, it was for me as well. Hopefully it was useful for the listeners. So thank you.